Now, here we are. And so where we left off, we talked about sex linkage and clinched the case that genes are physically located on chromosomes, and they have particular locations along a chromosome length. They're not stored just, any old where. But there is you could think of this as a little bit like, oh, you young post-millennial whippersnappers don't remember cassette tapes, I'm sure. But back in the old days when I was alive, you could get these cassette tapes of music, and they were long, reels of magnetic tape, and you'd play them in this thing that we had called a tape player, standing defiantly outside of your girlfriend's house. Not that I ever did that. And songs would play one after the other, and the first part of that long length of tape would carry, Hit Me One More Time, and then the next part of that tape would carry Oops, I Did It Again, and then the next part of that tape would carry Toxic or something like that, assuming we're talking about Britney Spears' greatest hits. And anyhow, so each portion of the tape corresponds to a song, and the songs are located at specific locations along the length of the tape. And genes evidently do something similar. But how does that actually work? Well, let us commence finding out. So we'd stopped with Sturtevant working out how to, hang on a sec. We'd stopped with Sturtevant working out how to do these linkage maps. So he worked out a way he could physically localize genes along a chromosome. But what is it about chromosomes that makes it possible for them to do to London and a bacterium? Griffith is studying a bacterium called strepbacterial pneumonia. These are, you might have seen this picture before if you remember. These are some Streptococcus pneumoniae that have been stained with probably just India ink, and you'll notice that they're surrounded with these light-colored halos. And the reason that is that the cells are surrounded by a capsule. If you go way back to about lecture 3, some bacteria, this mass of surrounding the individual cells, which are stained dark here. And the reason this matters is that bacteria that have the capsule cause very severe illness. Bacteria that don't have the capsule don't. Bacteria with the capsule can evade the body's immune system, and bacteria that don't have the capsule get mopped up by the immune system pretty quickly, and they don't make you especially ill. So having the capsule or not is the difference between a potentially life-threatening infection and something you might not even notice. Now, when we grow bacteria in the lab, we make this. It looks a little like Jell-O, although it's technically not. We make this gel substance. It's called agar, and it looks like firm Jell-O. And bacteria will grow on agar, but not in it. They can't break it down. They can't penetrate into it. They'll just grow up on the surface. We can put different nutrients and things, sugars and vitamins and what have you, that different bacteria need. If you ever do any medical laboratory work, you'll handle an awful lot of dishes of agar. I used to work with this stuff all the time when I worked at the Berkeley Free Clinic. And when a bacterium is placed on agar, it'll grow and divide and divide and divide and divide. But most bacteria cannot move over agar, although a few can. But the upshot is that what you'll get is a giant blob of bacteria called a bacterial colony. And S. pneumoniae that doesn't have a capsule will form colonies that are very small and look kind of rough. And that's called type R, which stands for rough. And bacteria that do have capsules form much larger colonies because the cells are all covered with slime. The entire colony looks glossy. And this is called S-type, which stands for. And so the R strain is benign. The immune system will mop it up pretty quickly. If you get R-type strep pneumoniae in your lungs, the only problem is it might turn you into a pirate because they're R bacteria. That was a joke. Yeah, your body will clean it up. Smooth ones that have that capsule of slime enable the bacteria to dodge the immune system. You can't fight it off nearly as well, and you end up with a severe illness. That's the S type. Well, Griffiths was doing some experiments on these bacteria, and he found that if you injected S-type bacteria into a mouse, you'd kill it within 24 to 36 hours. And then once the mouse was dead, you could take samples of the dead mouse's blood or other body fluids, grow them up on a dish of agar, and you'd get S-type bacteria. Okay, that's not unusual. You inject smooth bacteria into the mouse dies, you get smooth bacteria out of the mouse. If you injected R-type bacteria, the rough bacteria that don't have that slime coat, the mouse stays healthy. And if you then take a sample of the mouse's blood, you might get a few R-type bacteria out, but most of them are destroyed by the mouse's immune system. Most of those bacteria just get mopped up by the mouse's white blood cells. Okay, no real surprises there. Here was the surprise. You could take those S-type bacteria and kill them by heating them. And if you injected dead S-type bacteria into a mouse, the mouse stayed healthy. And if you took a sample of the mouse's blood and spread it on a dish of agar, you didn't get any bacteria back, right? The bacteria didn't grow inside the mouse's body because they were dead. Okay. None of this is unexpected, but here's what stumped Griffith. If you mixed living bacteria that didn't have the capsule, the R-type, and dead bacteria that did have the capsule, the S-type, so living R's and dead S's, if you inject that combination into the mouse dies. And then if you sample the mouse's blood and body fluids, you get live S-type. So you inject in live R and dead S, and you get live S. What? Yeah. Dead S doesn't kill the mouse. Live R doesn't kill the mouse. Mix the two, you do kill the mouse, and you get live S out of the mouse. Griffith reasoned that there must be something inside those dead bacteria. Since dead bacteria don't actually come back to life, I mean, you might be thinking of endospores, but these bacteria can't do that. So since the dead bacteria don't come back to life, he reasoned that there must be some substance, something, some chemical or something inside the dead bacteria that the live R bacteria were able to take into their cells, and that something turned them into S bacteria. So live R bacteria could become live S bacteria if they picked up some chemical from the dead S bacteria. So he reasoned something had passed from the dead bacteria to the living ones that transformed them, transformed their phenotype, and it transformed them from being harmless to harmful, and he referred to this as the transforming principle. Okay, he doesn't know what it's called. This is just a name we're giving it. So that raises the question as to what exactly was the transforming principle. There were various experiments to try to figure out what it was. I can't go into all of them. An American guy by the name of Oswald Avery and colleagues of his began growing lots and lots of Streptococcus pneumoniae, breaking them down into their macromolecules, separating different macromolecules out, and then trying to test which one was the transforming principle, which one would transform R-type bacteria into S-type bacteria. Took them 15 years, but they finally isolated and clinched the case as to what the transforming principle actually was. So what is this stuff that can pass from dead killer bacteria to live harmless bacteria and create live killer bacteria? It turned out to be a substance rich in phosphorus and nitrogen, as well as carbon, hydrogen, and oxygen, that had already been discovered back in 1869 by a Swiss guy named Friedrich Miescher. And he discovered it in the nuclei of eukaryotic cells, but it turned out to be in prokaryotes as well. So in eukaryotes, it's found in the nucleus. It's a weak acid, and Miescher called it nucleic acid, sensibly enough. And it turns out that one of the substances that chromosomes are made of is nucleic acid. The other one is protein, specialized types of protein. We'll talk more about protein later, but Avery was able to show that the protein was not the transforming principle, that the nucleic acid was the transforming principle, that nucleic acid was what was passing from the dead S bacteria to the live R bacteria and transforming them into live S bacteria. Okay, so what the heck is nucleic acid? Very, very briefly, matter is made up of tiny particles called atoms. There are 88 naturally occurring types of atom, or in other words, there are 88 naturally occurring chemical elements. There's also 30-some elements that have only been made in laboratories. We don't really need to talk about them here. We've got perfectly good chemistry and physics departments if you want to know about them. Atoms can form bonds with each other, and when atoms link together, they form molecules. And, when you bond multiple atoms together in a regular way, you get what we call a chemical compound. So when atoms stick together, they form molecules of a chemical compound. I'm not going to get into what atomic bonds are. Most of the time, for what we need to do here, we're going to represent atoms as little letters or possibly colorful ping pong balls. You probably already know that atoms are themselves made up of protons, neutrons, and electrons. You may know that sharing electrons is what makes chemical bonds able to form. When you get into chemistry, you know that it's a lot more complicated than colorful ping pong balls. But for what we need to do here, we're going to pretend that atoms are little balls that stick together with Velcro. It's not an accurate model, but it will do for what we need to accomplish in this class. And we can represent molecules like this. This is a molecule of a compound called styrene. You have a six-sided ring of carbon atoms that are stuck together with single and double bonds. Actually, they're stuck together with something a little fancier called resonance bonds. I'm not going to worry about that. One of the carbons is stuck to a group of two more carbons, and all of the carbons have hydrogens stuck onto them. This is just what this is called. This is called styrene. By itself, that's not so interesting, but you can take several styrene molecules, line them up like this, and link all of them together in a chain. And you can make these carbons bond with each other. I've drawn the new bonds there in blue. So all of these styrenes can be made to stick together into a very long unit, analogous to the way that lots of train cars can be linked together to make one train. When you link train cars together, you get a train. When you link styrene molecules together, you get polystyrene. Probably best known in the form where they blow lots of air bubbles into it and make styrofoam. You've handled polystyrene all your life. Plastics, all plastics are polymers. Polystyrene is one you might know. PVC is, the stuff they make a lot of pipes out of. PVC stands for polyvinyl chloride. Polyethylene is another. I can't even remember all of the plastic types, but what they all have in common is that they are organic molecules that are strung together in long chains. And that tends to make them solid and durable, which is why we make so many things out of plastic. Many biological molecules, including proteins and nucleic acids and many carbohydrates, are also polymers. Any large molecule that's built up out of subunits, repeated, arranged in a long chain, one after the other, is a polymer. Any molecule that has that kind of structure with a bunch of small molecules that are stuck together. And you could compare polymer molecules to trains, where you have a train that's made up of one type of car, or possibly just a few types of car, strung together in many possible combinations. Some polymers might be made of only one type of monomer. A monomer is the simple molecule that you string together. Something like polystyrene would be made of only one type of monomer, like a train with only one type of car, maybe a train with only boxcars or something. But many polymers, including nucleic acids, can be made of several different types of monomer, like a train that would consist of some boxcars and some flat cars and some tank cars, or something like that. And, right, just like you could string together cars of many different types on a train, tankers and boxcars and flat cars and flatbed piggyback trailers and, who knows what. You could string those in combinations that will carry almost any load you can think of. Polymers are the same. There are some polymers that are made of only one type of monomer, but there are other polymers that can be made up of several different monomers strung together in any conceivable order. Okay, here's a biological polymer. These things that look like hexagons are rings of carbons and hydrogen atoms, and then O stands for atoms of oxygen. So each one of these hexagons with O's stuck onto it represents simple sugars. And a simple sugar in this case is a ring of carbons and hydrogens with some oxygens thrown in for flavor. String a whole bunch of simple sugars together and you'll get starch. String them together in a slightly different way and you get a very tough molecule called cellulose. You might recall that cellulose is what makes up plant cell walls, that makes up, things like wood and paper, which we make out of plant cell walls. Chitin happens, you might remember that as the cell wall of fungi. That's another polymer. That's a polymer of slightly different sugar molecules that contain some nitrogen too. Proteins, which I know you've heard of, are also polymers of simple molecules called amino acids, which we will get to soon. So polymers are extremely important in organisms. And nucleic acids turned out to be polymers as well. Well, what are they polymers of? What are the cars that make up that train? It was worked out in 1919 by a guy named Phoebus Levine. And he worked out that the monomer, right, the simple molecule that is strung together to make up nucleic acids, is called a nucleotide. And this is a nucleotide. I should tell you that carbon atoms are so common that we often don't draw them. So anywhere you see an intersection of lines that doesn't have a letter there, there should be a carbon atom there. We just usually don't bother to depict it because carbon is so ubiquitous. So everywhere you see a two or three or more lines coming together at a point, that point represents a carbon atom. So nucleotides make up nucleic acids the way that cars make a train. And just like a train car itself is going to have parts like the wheels and the base and the couplings and all that, just like a train car is made up of parts, a nucleotide is itself made up of parts. Drawn here on the left, you can see there's an atom of phosphorus, abbreviated P, surrounded by 4 oxygens and a couple of hydrogens. That's called a phosphate group. And the phosphate group is stuck to a ring of carbon atoms with some oxygens thrown in. That happens to be a simple sugar. And then the sugar is attached to in this case, it's a double ring of carbon and nitrogen atoms. Remember, the carbons are everywhere you're seeing an angle, even though they're not drawn here. So you have a ring, and in this case, it's a double ring of carbons and nitrogens, and we refer to that part as the base. Okay, and there are several bases used in nucleic acids. The Fender Precision, Squire, Ibanez, and Fender Jazz. Actually, I'm kidding. I'll talk about the bases in a minute. In the nucleic acid that we'll be discussing right now, the one that's most important, the sugar molecule is missing an oxygen atom that would normally go here. If there was an oxygen present sticking off of this carbon right there, the sugar would be called ribose. But that oxygen is missing, so this is ribose without an oxygen, aka deoxyribose. So the sugar is the sugar deoxyribose, and the name of the polymer, what you get when you take a whole bunch of these nucleotides and string them together, is deoxyribonucleic acid, or DNA for short. I'm not going to require you to memorize deoxyribonucleic acid. Well, you probably should, but you can usually just get away with writing DNA. That's what that stands for. And DNA turns out to be that transforming principle. The reason why those harmless bacteria were turning deadly is that they were picking up DNA from the deadly bacteria. DNA is a polymer of nucleotides, and all nucleotides have the same phosphate group and the same sugar group. All right, they've all got a phosphate group, they've all got a deoxyribose sugar, but they have 4 different nucleotides that are possible. If we go back to a train, think about a train where all of the cars have the same wheels and the same suspension, right? Because they've all got to ride on the same track. So they've all got the same wheels, the same suspension, the same couplings that hook them together. But what's on top of all that might be either a boxcar body or a tank car body or a flat car body or an Amtrak superliner body, whatever it might be. So all of your cars on a train are going to have the same wheels and the same suspension and the same couplings. They'll differ in what's on top. Same kind of thing here. All of these nucleotides have a phosphate group and a sugar group, but they can have one of four possible bases. And the base is either a single or a double ring of nucleotides. And the bases can be adenine, thymine, guanine, or cytosine. We can usually get away with just abbreviating those as A, T, G, and C. Okay, so like making a train out of four different types of car, you can make DNA from four different types of nucleotide, and what makes them different from each other is that they'll have one of four bases. So those bases are extremely interesting. In fact, DNA scientists are fond of singing, "I'm all about the base, no phosphate, I'm all about the base, no sugar, I'm all about the base." Okay, sorry, I'm just imagining you laughing uproariously here, so let's just mercifully move on to the next slide. Right. Okay, I promise we're almost at the peak right here. This is a lot to absorb. In the 1940s, a biochemist by the name of Erwin Chargaff discovered some facts about DNA. One of the things that he found was that the composition of nucleotides was different in different species. So human DNA is about 29% adenine nucleotides. Octopus DNA is about 33%, corn DNA is about 27%, and so on. For your purposes, there's no really interesting reason why. It's just a number, but it does tell you that different species have DNA with a different composition, and you might expect that. That doesn't sound too surprising. But when he broke it down, it turned out that the percentage of adenine was always equal to the percentage of thymine. So if your DNA contains 29% adenine, it also contains 29% thymine. And the percentage of guanine always equaled the percentage of cytosine. And the percentage of adenine plus guanine always equaled the percentage of thymine plus cytosine. You can show that with some simple algebra. So if you know one, you can calculate the other three. Think about it. If your DNA has 29% adenine, it also has 29% thymine. Add those up and you get 58%. Okay. The amount of guanine plus cytosine is 100 minus 58%. That's 42%, right? And if guanine equals cytosine, then the amount of guanine in your DNA must be 21%, because that's half of 42. And the percentage of cytosine is also 21%, because it's half of 42, and it's equal to the amount of guanine. And the percentage of adenine plus guanine is 29 plus 21%, which is going to be 50. And that always equals the percentage of thymine plus cytosine. We still call these Chargaff's rules. There will be a question or two on the test where you have to apply them. It's basic algebra. Just remember that the percentage of adenine equals the percentage of thymine, and the percentage of guanine equals the percentage of cytosine, and you can work everything out from there. Right. So what? Early 1950s. On the left, British scientist Francis Crick, which rhymes with prick, and did I say that? And American scientist James Watson, who's come over to England, and they're working on DNA at Cambridge University in the lab of a senior scientist named Maurice Wilkins. Crick and Watson you do kind of need to know. Those are names I might test you on, conceivably. And here's one I will test you on. Also working in Wilkins's lab was, yeah, I now get to talk about a female scientist, and a very good one, a woman named Rosalind Franklin. And Franklin was one of the world's best in a lab technique called X-ray crystallography. Okay, what the heck is that? In X-ray crystallography, you take, okay, my grandmother used to have these window ornaments that were just pieces of crystal. They were called sun catchers. And she'd hang them in a bright window, and when the sun would shine through the window, sunlight would hit that crystal and it would be refracted, meaning it would break up into a whole bunch of sunbeams that would make little spots of light on the walls. She also had a crystal chandelier in the dining room, and when she turned on the light from the light bulbs would pass through the crystals on the chandelier, and each crystal would break up rays of light into a bunch of little spots of light, which was pretty. Maybe your grandparents have something similar, or you've seen something like this. A beam of light hitting a crystal will break up into lots of spots of lots of little spots of light. It works with other types of energy. And in X-ray crystallography, you make a tiny crystal of some substance, whatever it is, and you zap it with a focused beam of not visible light, but X-rays. The crystal will break up the beam into many little beams, which you can detect not with your eyes, but with a sheet of photographic film. And you end up with something like this. You end up with a pattern of dots. And if you measure the positions of the dots on the film, you can see there's a regularity to it, right? They're not just randomly scattered. They're forming these concentric rings. And there seem to be kind of fixed numbers and a standard spacing of these dots in the ring. If you measure the positions and the angles of the dots, it is possible to work out the position of the atoms in the crystal. So even though atoms are far too small for you to see with any microscope that you're ever likely to work with, you can work out where they're located, what their positions are, what their distances are from each other, what the angles are that they make with each other. You can work all of that out by measuring the positions of the dots on the film and then applying some rather difficult and complicated mathematics. This is not easy to do, and Franklin was one of the best in the world at this technique. Okay, this is one of the ways in which we know where those atoms are located and how to draw those dots and lines that you've seen is X-ray crystallography. Franklin didn't invent it. That was invented back in the 30s, but she was very good at it. And this is one of the most famous photos in science because she made a crystal of DNA, which was not easy to do, zapped it with a beam of X-rays, and got the picture that you see on the right. Now, you can see that you have a pattern of dots that make an X. That X showed that the DNA molecule, if you could see it, was shaped like a coil, or as we call it, a helix. A helix is any kind of coil or spiral. A bed spring is a helix. A corkscrew is a helix. A spiral staircase is a helix. A snail shell is a helix. And the DNA molecule is a helix. That photo shows a pattern of dots that's typical of helix-shaped molecules. She was even able, by measuring the positions of those dots, to work out how the atoms were spaced, how wide the helix is, and how tightly it's coiled. Whether it's coiled so that the coils are practically touching or whether the coils are strung out. she was able to work out what those proportions were. Okay. Watson and Crick got hold of Franklin's data, be it said without her permission. So they knew that the DNA molecule had to be shaped like a helix. They knew how wide the DNA molecule was. They knew how tightly coiled it had to be. They were able to work out that however it was arranged, the phosphate groups on the nucleotides were on the outside of the helix. They knew how big nucleotides were. They knew what their shapes were. And they knew about Chargaff's rules because Chargaff had visited the lab a couple of years before. So Watson and Crick sat down with a bunch of models of nucleotide molecules, basically little balls stuck together with sticks, and tried to come up with models that would fit all of these criteria. We know DNA is made of nucleotides. We know how big those are. We know their shapes. We know how they fit together. We know that whatever arrangement that they're in must satisfy Chargaff's rules. We know that the finished product has to be a helix, and it has to fit certain dimensions. Put all of that together, and on April 25th, 1953, Crick, who's pointing to it, and Watson down below, who's looking kind of stunned, published a paper that described their model for the shape of DNA. Not one helix, but two chains of nucleotides, two polymers wrapping around each other like two threads wrapped around each other, forming a structure called a double helix. Not a single helix, a double helix. That's two different ways of representing a double helix in 3 dimensions. On the left, that's a stick model where the atoms are just the corners of those sticks joining together. On the right, a space-filling model where the atoms are represented as little ping pong balls stuck together. They're both just models. They're not exactly what your eyes would see if you could somehow see a DNA molecule, but they'll give you some idea of what we're talking about. The sugars and the phosphates in both of these pictures are colored white and pink, and the bases are kind of on the inside of the double helix, and those are colored red and yellow. So that is kind of what your eyes would see if you could somehow shrink yourself down small enough to view a DNA molecule. If you could take a piece of DNA and untwist it, it would look kind of like a rope ladder. In this diagram that I've got here, the sugars are those pentagons that are colored a very light green, and the phosphates are kind of a tan color. And DNA is made up of 2 strands. What holds the strand together is bonds from sugar to phosphate to sugar to phosphate to sugar to phosphate. Those make up the sides of the ladder. If you look at the left side and the right side of that diagram, you can see that they're made up of phosphate stuck to sugar, stuck to phosphate, stuck to sugar, alternating as you go down either side of the diagram. The bases point inwards, and they form a type of chemical bond with each other to form the rungs of the ladder. So we've got a kind of ladder-shaped molecule. If we let this twist, the ladder will twist into a spiral, and we get this double helix. And here the bases are shown as those groups of ping pong balls. For clarity, the sugars and phosphates are just shown as a couple of ribbons. They're not shown as individual molecules, but hopefully that will help you see the double helix. And in fact, we can actually confirm that Watson and Crick were basically right. On the left, you've got a model of the double helix. On the right, There's a technique we can use called scanning tunneling microscopy. We don't need to know about how it works. As far as I know, we can't do it at UCA. It is it takes very expensive equipment and expertise. But back about 20 years ago, somebody actually was able to put some DNA under a scanning tunneling microscope and take a picture of it. And, it's a little rough, but you should be able to see that it actually does look like that double helix. You can see the sugars and phosphates on the outside and then the bases on the inside. So it's actually been possible to confirm that model. Okay, little bit of a soapbox here. Watson, Crick, and Wilkins won the Nobel Prize in 1962, the Nobel Prize in Medicine. Many people feel that Roz Franklin should have shared the prize because she was the one that provided that critical data on, she was the one who showed that DNA was a helix and showed what its dimensions were, how wide and how tightly coiled it was. Sadly, she died in 1958, and they don't give a Nobel Prize posthumously. It's only given to a living scientist, although there have been a couple of cases where a scientist has been living when they awarded the prize, but died before the ceremony where he'd actually get it. And in that case, the prize is given to his heirs. But you can only be awarded the prize if you're still alive. So she could not have shared the Nobel. So technically, but her importance to the story has often not been recognized. And that's in part because James Watson wrote a book called The Double Helix. It's worth reading because it gives you a look into the mind of a very intelligent scientist at the top of his game who is working on something that he knows is going to be critically important. Because the shape of the DNA molecule is the key to understanding how it works and how it does what it does, how it is that it can carry genes, right? That ultimately is going to come down to the shape of the DNA molecule. Watson knew he was onto something really big. This makes him one of the most famous scientists in the world. And he published a book in 1968, The Double Helix, that basically gives you a great look into what it was like to be, young and hot shit and on the trail of something that is going to blow open biology. But he's so focused on himself that he spent most of the book downplaying Rosalind Franklin. Much of what he wrote about Roz Franklin and the double helix was complaining about the way she didn't dress nicely. She was not unattractive and might have been quite stunning had she taken even a mild interest in clothes. This she did not. There was never lipstick to contrast with her straight black hair, while at the age of 31, her dresses showed all the imagination of English blue-stocking adolescents. Yeah. She doesn't dress nice. She doesn't smile. So he kind of writes her off as not being very important to the story. Yeah, sexist AF. First of all, that wasn't actually true. Roz Franklin never married, but she did enjoy the theater. She did dress up very nicely to go out with friends. She had an active social life, and she could be a wonderfully warm person. However, she didn't suffer fools gladly. When she was on the job, she was very professional, very proper, and Watson, to be frank, was a bro. I'm not sure what frat Watson was in, but I'm sure he was in one because that's the kind of guy he was. And it's unfortunate that this remains a problem for women today. If you try to act professional and proper, you may get dinged for being a cold bitch. If you try to be nice and kind in the workplace, people are going to tend to walk all over you. That tends to be a dilemma that still besets women in the professions today. I would hope it's better, but it certainly has not gone away. It's the kind of bind that a lot of women find when they try to move into professional circles. Some people will treat you either as a bimbo or a battle axe, and there's no real in between. And Franklin definitely went with the battle axe option. She was very professional, didn't go around, smiling a lot or giggling or making tea or anything like that. She was all business, and Watson tended to write her off because of that. I've heard female professors in my department claiming that when they get course evaluations back, there's always some bros that write things like, "You should smile more," which is basically irrelevant. But this kind of double standard still does persist. I would hope it's gotten better, but it certainly has not gone away. She does deserve a large share of the credit. Watson did later acknowledge her accomplishments. So here he is today, not looking like such a bro, admitting that we both came to appreciate her personal honesty and generosity, realizing years too late the struggle that the intelligent woman faces to be accepted by a scientific world, which often regards women as mere diversions from serious thinking. So Watson did eventually acknowledge that he had been unfair towards her. If we could get him to stop spouting racist shit, it would be even better. He's got kind of a very bad track record where that's concerned, and he's being called out on that. At least he's repented of his sexism, and that may be the best we can hope for. So, yeah, not a perfect man by any stretch of the imagination, but one who does get some of the credit for a pretty big discovery, and too late acknowledge the help that he got from Ross Franklin. Right. So Watson and Crick, with Franklin scoring an assist on the play, worked out that DNA is shaped like a double helix. Why should you care? The reason is that to understand how objects work, you have to understand their shape. Right? Think about tools. The shape of a hammerhead, or in this case, the shape of a screwdriver head, determines what that tool can do, right? The Phillips head on the screwdriver determines what kind of screw the screwdriver is useful for screwing and unscrewing, right? Hammers have different shaped heads depending on whether it's a claw hammer for driving nails or a blacksmith's hammer for hammering hot iron, or a sledgehammer for driving posts into the ground, or something like that. To understand how an object works and what that object can do, you have to understand their shape. And understanding the shape of DNA is the key to understanding what DNA does and how DNA does it. That shape of DNA is one of the big secrets of life. And now will be a very good time for a break for now. And there's me. So I will stop presenting and I will stop recording. And we'll pick this up in, I've got 11:06. We'll pick this up at 11:20.